Hello everyone, today I would like to present our research with title Vegetation Analysis and Diversity of Seed Plants or Spermatophyte in the area around Porok Beach, Yogyakarta. I would like to tell you about the outline. First, introduction. Second, method. Then third, result and discussion. Fourth, conclusion. Introduction. Preservation of spermatophytes is not only about how to care for them, but also in the identification. The diversity of plant species greatly affects the stability and sustainability of nature. To find out the diversity of plant species, data on the composition of vegetation is needed, including the structure and composition of vegetation. Vegetation analysis must be carried out so the biodiversity of a location can be known and to facilitate maintenance and empowerment. Vegetation analysis requires type and quantity data to determine the important value index in order to obtain quantitative information about the structure and composition of plant communities. This research aims to determine the diversity of seed plants around the area of Porok Beach, Yogyakarta through vegetation analysis. Method, place and time of research in the area around the Porok Beach, Yogyakarta. Wednesday, July 13 to 14. Research method, population and sample. Population all seed plants, spermatophyte, found in the area around Porok Beach. Sample, seed plants with 3 habitus, plot namely the quadrat transect with a size of 10 times 10 meters, taken by purposive sampling. Research type and designs. The transect and quadratic methods are used in this descriptive and quantitative research. A transect line along 70 meters in the area of seed plant vegetation with 3 habitus. 3 plots are squares 10 meters times 10 meters and the distance between plots being 10 meters. Repeated 3 times equal 9 plots. Tools and materials. Tools, pH meters, pegs, roller meter, sewing meter, stationery, camera, laptop, and mobile phone. Materials, rope, Ziploc plastic, identification books, observation sheet, and plants that have been found and identified. Result and discussion. Identification of seed plants, 3 habitats in the area around Porok Beach, Yogyakarta. First, Casuarina equistifolia, the species suitability for planting as windbreaks in based on its finely branched crown and dense canopy of horizontal branches. Shade trees are suitable for protecting sandy soils from erosion. Second, Terminalia catapa, a spreading fibrous root system that plays an important role in the soil stabilization. This tree is tolerant of strong winds, exposure to salt, and moderate high salinity in the root zone. Third, Pandanus odorifera. This dioecious tree has small palm-like branches that usually grow in the tropics and some warm climates around the coast. Fourth, Cocos nucifera. It has strong roots. It cannot penetrate hard soil. The last, Panthis acidula. Single leaves and grows crosswise. Result and discussion. Dominant and codominant families of seed plants, tree habitus in the area around Porok Beach, Yogyakarta. The result of research conducted in the area around Porok Beach, Yogyakarta from 9 plots found as many as 5 species of seed plants, namely Casuarina equisetifolia or Seaweed, Terminalia catapa or ketapang, Pandanus odorifer or sea pandan, Cocos nucifera or coconut, Pampis acidula or stigi, from the families Casuarinaceae, Combretaceae, Pandanaceae, Arecaceae, and the last, Litraceae. Parameter of vegetation of seed plants, spermatophytes, tree habitus structure in the area around Porok Beach, Yogyakarta. Based on the table, it can be seen that vegetation structure of seed plants with the highest tree habitus was Terminalia catapa, while the structure of seed plants with the lowest tree habitus was Pampis acidula or stigi. Terminalia catapa or ketapang was indicated to have high adaptability so that it can survive, preserve its species, and dominate in a community. Result and discussion. Diversity index of seed plants, tree habitus in the area around Porok Beach, Yogyakarta. Vegetation of seed plants, tree habitus in the area Porok Beach, Yogyakarta has a moderate diversity index value of 1.49, still ranging from 1 to 3. The level of diversity of seed plants, tree habitus in the area around Porok Beach, Yogyakarta is indicated to be influenced by soil organic matter, soil moisture, soil pH, temperature, humidity, and light intensity in the area. The diversity of seed plants that has been carried out in the area around Porok Beach, Yogyakarta, with tree habitus is also supported by abiotic factors, namely soil pH of 7. In the general, the area Porok Beach, Yogyakarta includes both straight and rocky beaches. Conclusion. Based on the result of the research, it can be concluded that there were 5 families, 5 species, and 33 individuals of seed plants in the area around Porok Beach, Yogyakarta, including Casuarina equisetifolia, Seaweed, Terminalia catapa or ketapang, Pandanus odorifer or sea pandan, Cocos nucifera or coconut, and Pampis acidula or stigi. The dominant sea plant Spermatophyte in the area around Porok Beach, Yogyakarta was Terminalia catapa or ketapang with an INP value of 0.85%. The diversity of seed plants or spermatophytes in the area around Porok Beach, Yogyakarta is classified as moderate, which is 1.49. I think that's all for me. Thank you for your attention.